dear students. So let's talk about cell membrane. So guys, what is a cell membrane? This is a protective layer, which is separating the contents of the cell from the extracellular environment, right? Now guys, the question on the cell membrane is mostly like the fluid mosaic model of the cell membrane, right? That is the most common question you will be getting in your exams. Fluid mosaic model. So what is the meaning of that? So guys, this was proposed by Singer and Nicholson, and the meaning of mosaic here simply means that there are multiple things are involved to make the cell membrane, right? Guys, the cell membrane in the eukaryotic cell or the plasma membrane is a lipid bilayer, right? That's a lipid bilayer, and this lipid has also proteins, right? It has phospholipids, it has cholesterol, it has carbohydrates or the glycolipids or the proteoglycans are attached to it. So basically, all these things, lipids, carbohydrates, and proteins together, they are making a mosaic appearance, right? Mosaic appearance means what? That means multiple things present together, not a clean appearance, right? I'm going to select that mosaic appearance. Fine. So let's talk about the lipids first of all. So guys, if we talk about the lipids in the cell membrane, it's a phospholipid bilayer. Cell membrane is a lipid bilayer, and the most common lipids are phospholipids, right? Glycerophospholipids, actually, I would say, right? So, what are the types of glycerophospholipids present here? Now, first of all, look at the structure of phospholipid. It has a hydrophobic domain and a hydrophilic domain. The hydrophobic domain, right, is made by the fatty acids mostly, and the hydrophilic domain is made by the alcohol and the phosphoric acid. So as you can see the structure here, you can see that the polar groups, right, that is hydrophilic portion of the phospholipids, that is pointing towards the outer area, that is extracellular fluid, and that is also pointing towards the cytoplasm. So as you can see, this is how we are getting the lipid bilayer. So it is arranged in such a way that all the hydrophobic portions are present together, right, towards the membrane. Hydrophilic portions toward the cytoplasm and extracellular fluid. So that is like the basic structure. But guys, this lipid bilayer is asymmetric in structure. Means it is a different composition of the lipids towards outside or the outer layer, and the inner layer, they have different composition. Overall, the most common phospholipid is phosphatidylcholine or the lecithin, right? So lecithin being the most common phospholipid overall, and guys, that is most commonly present in the outer leaflet of cell membrane. In the inner leaflet of cell membrane, right, which is facing towards to the cytoplasm. So the inner leaflet of cell membrane has more commonly phosphatidylserine and phosphatidylethionelamin, which is also called as cephalin, right? So these two lipids are more common towards the inner side. That's why we say lipid bilayer or the plasma membrane has an asymmetric distribution of lipids. It's asymmetric in nature. Correct, guys? And guys, it is also found that during the cell injury, if this phosphatidylserine is exposed, to the extracellular fluid, then that's a signal of cell death, okay, or the apoptosis. Fine. So guys, that is one basic thing. Now, some other lipids which are found in the cell membrane is sphingomyelin, which is a type of sphingolipid. Guys, remember one thing, right? That triglycerides are also the lipids, but they're never found in cell membrane because they're so simple lipids. So they're not found in the cell membrane. Now, the other thing which is found in the cell membrane is cholesterol. Because cholesterol and phospholipids both are amphipathic in nature. That means hydrophilic and hydrophobic. How the cholesterol is amphipathic? Cholesterol has a OH group, hydroxyl group, right, on the third carbon. So that will be placed in such a way that it is, towards the water side. Got my point? Okay. Now, still talking about lipids. Guys, the fluidity of the membrane is determined by many factors. And cholesterol is also one of the most important factor in them. So, cholesterol content decides the membrane fluidity, right? Fluidity means, as you can understand, right, how much it can be in the fluidic state instead of a more packed or crystalline state, right? So, below the transition temperature, the cholesterol increase the fluidity if it is high, and above the transition temperature, cholesterol decrease the membrane fluidity. So it has both the roles, right? Guys, what is the transition temperature? When the crystalline structure of the membrane, or the more packed or the more rigid structure of the cell membrane converts into more fluidic in nature, that is called as a transition temperature, right? Up to 60 degree, beyond 60 degree is called as transition temperature of the this membrane, that is plasma membrane. Fine, guys. Okay, so now moving to the next point. What else determines the fluidity of this fluid mosaic model? So the other thing which what else determines the fluidity of this fluid mosaic model? So the other thing which determine is the fatty acids. Guys, the fatty acids should be unsaturated. They increase the fluidity. Not saturated, unsaturated. Okay. Second thing is cis type of fatty acid should be present. They also increase the fluidity. Trans fatty acids decrease the membrane fluidity, but cis increase the fluidity. So three things which maintains or which changes the fluidity of the membrane. Okay, guys, that is your lipids. If you're talking about the lipids, guys, let's talk about proteins. There are two types of proteins which are found in the cell membrane: integral proteins and peripheral proteins, right? Integral proteins are the proteins which are traversing the membrane, right? So they are moving from the membrane or to the membrane and may go again and again. Like, for example, guys, integral protein, there is a glycophorin protein found in the RBC membrane, which is just going towards the cytoplasm just one time, single pass, right? Then, guys, we have a G protein called receptors. These are the proteins which are called as sometime proteins, and 7-pass transmembrane proteins. So they are passing through the membrane. Protein coupled receptors. These are the proteins which are called as serpentine proteins, and 7-pass transmembrane proteins. So they are passing through the membrane 7 times. As you can see here, 7 TM we call them, transmembrane proteins, which is going 7 times through the membrane. So what do you call this? The serpentine transmembrane proteins, or the G protein coupled receptors, that we will discuss again later many times. So guys, we have also the glucose transporters, and glucose transporters are, they are passing through the membrane 12 times, right? Okay, so we have various types of proteins, right, which can be transmembrane, which are mostly integral, and they are passing through the membrane multiple times, right? Then, guys, we have the peripheral proteins. Also, peripheral proteins are the proteins, which are, we can imagine like this way. As in the fluid mosaic model, what they said is that the proteins are floating icebergs in the sea of lipids. As you can see the structure, the extrinsic or the peripheral proteins, they can be visualized as the proteins, right, as the icebergs floating in the sea of lipids. Fine. What is the function of these peripheral proteins, guys? Guys, they can act as receptors, right? For example, if I say, receptors which are, helping to bind certain hormones. Fine. So these proteins can be just receptors, peripheral proteins. Okay, now, guys, if we talk about certain named disorders which are due to changes in the protein structures, or because I told you these proteins are acting as channels, acting as receptors, etc. Okay, so guys, certain receptors like aquaporins are present in our cell membranes, right? Especially, let's say, in the kidney collecting ducts, we have certain certain channels which are called as aquaporins. See guys, the cell membrane is permeable to water, right? But it is limited. So this permeability can be increased by certain proteins known as aquaporins, right? So these aquaporins, especially in the kidney, their levels are increased by certain hormones like ADH, antidiuretic hormone, and this helps kidney to reabsorb more water, retain more water. Okay, so that is why if there is a mutation in the aquaporins, right, so there will be less water reabsorbed and more water excreted, and this disease is called as diabetes insipidus. It's not the diabetes mellitus, diabetes insipidus we call it, right guys? Similarly guys, there is one more disease we will also discuss later again, that is called as cystic fibrosis. In the cystic fibrosis, there is a protein which acts as a channel, that is called as CFTR, cystic fibrosis transmembrane receptor. Fine, this channel has a property that it is helping to make the secretions of the glands more thin, right? It secretes chloride actually, right? So it's a chloride channel basically. So because of its defect, what happens, the glands, they're not able to secrete properly, and whatever they secrete, it becomes very rigid or very viscous, because the water content is lost because of the defects in chloride channels. So CFTR or cystic fibrosis we'll talk again later, right? So these are certain proteins that you should know in terms of disorder, right guys? Now guys, these proteins are also anchored to the cell membrane or the lipids by certain special modifications. Like we have certain modifications like isoprenoid chains, which is a type of a lipid, isoprenoids. Then we have GPI, right, glycosyl phosphatidylinositol anchors. So they are all helping in the attachment of the proteins to the cell membrane. GPI anchored enzymes are there, like for example acetylcholinesterase is bound to the cell membrane by the GPI anchors, glycosyl phosphatidylinositol anchors. Okay, for example, like this. Now guys, moving to the carbohydrates. Carbohydrates in the cell membrane, right, are found only in the eukaryotes, not in the prokaryotes. So they are present in the form of glycoproteins, right? Like insulin receptor is a glycoprotein. So sugar containing protein basically, I can say like this. Or they may be present attached to the cell membrane in the part of ECM. That's a part of ECM actually, and that is proteoglycans. Okay, so proteoglycans may be attached to the cell membrane. So basically guys, whatever the carbohydrate is present near the cell membrane, we call it glycocalyx, which are mostly proteoglycans. Or the cell membrane itself has certain receptors or channels which are glycoproteins, right? For example, like insulin receptors. Fine guys, so that is all about the fluid mosaic model of the cell membranes. Thank you. See you in the next video.